Hi, everybody, and welcome to today's Germination Retail Roundtable webinar. My name is Mark Zinkowitz, and I'm editor for Germination, and today I'm happy to be your host. Today's theme is bacterial leaf streak. We'll talk with 6 experts who will shed some light on what they're seeing out there in regards to this disease, how it works, what it looks like in Canada, and what's being done in the way of research. First, I'd like to thank our sponsors, 2020 Seed Labs and CCAN, for their support. This webinar is being recorded and will be made available at germination.ca following the proceedings. Questions are welcomed at any time during our webinar. If you have a question, please type it into the chat box and let us know who it is directed to. As I said, today's webinar is about bacterial leaf streak, which you're looking at right now. This disease has been making headlines as it becomes more prevalent on the prairies. Bacterial leaf streak is actually a pretty major disease of cereal crops around the world and can cause big reductions in yield in areas that are particularly hard hit. Major losses have not been reported in Canada as of yet, but the disease is on the rise in Western Canada and is cause for concern. We'll talk with 3 researchers who are doing some work in regards to this disease. But first, I talked with 3 people about what they're seeing out in the field right now. I spoke a few days ago with Eric McLean. He's a seed grower here in Manitoba. He's owner of J.S. Henry and Son. And I got to talking with him about his experience with bacterial leaf streak and what seed growers are concerned about right now. So I'm going to play our interview for you. It's about 9 minutes, so sit back and enjoy Eric's comments. Well, thank you for joining me today here, Eric. How are you today, sir?
Doing very good, Mark.
Excellent. So yeah, as you know, we're talking about bacterial leaf streak today, and I wanted to talk to you because I understand you've had some experiences in this department as a seed grower. Can you kind of recount for us what experiences you actually have had with this disease?
For sure. I mean, from what I've seen and what we've heard from others over the past year, as we review back, we look at last year's growing season was a good place to start when I started thinking about this. And we saw many potential perils which could have impacted us along the way, including a long winter, delayed thaw, wet spring soils, and delayed seeding. A lot of crop residue was left to rot as the fall of 2019 was wet, and harvest drew out and little field work was done. The spring dried out later by 2 to 3 weeks for some, and the crop went in relatively flawless as the crop was in. We waited and waited for moisture. And things were looking dry by late June, and many were worried about drought setting in. And then the drought was broke. I personally was on the edge of a significant rainfall event, which occurred right at flag leaf timing in the crop. And the moisture levels were increased 6 to 10 times the normal within a 72-hour period. And humidity shot up with average daily temperatures over and above 26 Celsius for the following weeks. And this sets the stage for our topic we're here today to talk about. And we saw what we mistakenly identified as a fungal infection, to which we spray fungicide on to control, but the crop failed to grow out of it. And many of our farmer customers started to call, worried. They asked what was happening and what could be done. I asked a few trusted advisors and researched what was happening, and uncertainty gave way to helplessness as we knew that we were in the infected area and came to understand that we could do little to salvage the situation. So bacterial leaf streak, or BLS, I might say, just to shorten things up a bit, or black chaff, as I've also heard it called, is present in growing areas around the world where moisture and warm growing conditions are plentiful, often under irrigation. It's not something that Western Canadian varieties are screened for in development, as we can often be arid or definitely drier and cool enough for it not to be a problem. So it can cause damage at various stages of development, as it's polycyclic, which meaning that infection can occur throughout the growing season, starting from overwintering on host plants, infected seed, soil, wind-borne infection, which all combine with the right conditions and can lead to rapid development. There are no completely effective treatments out there, and some treatments have shown some effectiveness through time, but nothing is better than genetic resistance. And the best way to manage the disease is to source clean seed from unaffected areas and growing varieties that have expressed resistance in trials. But with no official ratings given to Canadian varieties upon registration, that part is difficult. So knowing these handful of facts and comparing it to what happened to farmers in the Eastern Prairies, the picture becomes clearer as to why bacterial leaf streak impacted so many acres of a popular variety, which made up to about, personally, 20 to 30% of our sales over the past few years.
Are you hearing a lot of concern from other seed growers about this particular disease?
As it's a tough call, because from a seed grower perspective, I've managed to sit in many circles, and one of the things that I'm part of is I'm a member of the disease evaluation team for the Prairie Grains Development Committee, and that grants registration of new cultivars in Canada, registration for them. I'm reserved about how much we have to worry about this and how much lasting damage this disease will have. It doesn't mean that this threat is gone, but we must be, we've got to be practical. And as I've stated, we've had a very dry fall and winter, and moisture levels are quite low. We have a dry year, and the disease cycle will be broken, hopefully, or significantly reduced. I'm hoping. Many other varieties in production express some or very good resistance to bacterial leaf streak, and sales of infected varieties are essentially non-existent. Farmers are very shrewd, and they don't have room in their rotation for risks. So in many ways, the market has imposed all the cure that it's needed. And the worst thing that could happen is that governments, the seed industry in general, gets involved and restricts sales of infected varieties, in my opinion, because the market will do a lot of this work for it. I wanted to just put out there, I did not clean my infected production, and I didn't do any testing on it because of the market situation. There was no point in cleaning it if nobody was going to be buying it. So I don't know how much, BLS would or could have been removed in the cleaning process over the winter. That's one of the things we didn't get a chance to play around with. So as seed growers, how much of this is going to be an issue is always the question we come back to. So, but just as fusarium head blight has become a problem in Western Canada, we have learned to live with it. It became a great concern, and controls and breeding efforts became effective, and now it's easily managed. And so a lot of these things have to be taken into stride, as to say what is necessary and what is needed to be done. I don't know that I'm really big on how much restriction can be enforced with little benefits when the risk is so low. Was this just a one-off is what I'm getting at. How much will we have to look at for infection rates going into this next season? So we unfortunately need to have another year or so of this, I would say, affecting other varieties to take proper notice and to elevate it to a priority. If it fails to register as a problem, and so be it. If it becomes back with some more potency and starts to affect other varieties, then we'll have a direction, and then we'll have a base for applying funding and resources. In the meantime, I know that weed breeders do the best they can to give us great performing varieties, and as time goes on, I hope that they will be granted that knowledge and the tools to be able to satisfy the needs of farmers. But the trouble is that the needs change as soon as the solution comes for yesterday's problems. Sorry about that. I can never tell when my phone's going to be muted out or what I'm going to have to do. So hopefully that's quiet now.
Not a problem. That's the virtual world for you.
Yeah, that's
What
right.
are you looking for in terms of information about this disease? Because we'll be talking with some researchers on today's webinar who are looking at the disease, how it works, what could potentially be done about it in the way of testing, etc. What information do you feel is lacking right now that you could really use as a seed grower?
It's just in trying to prepare for this webinar, I've done some homework on the topic, but I'm no expert, and I usually try to logic things out by question and answering in my own head. So bear with me. So because I'm thinking that I'm sure after hearing the other pathologists and experts, farmers, we'll all have heard the same thing here, and I'm thinking that at the end when we balance things off against what we do know, which is that dryness, lower levels of crop residue, extended or consistent crop rotation, and clean seed will go a long way to reducing, the harm from BLS. But looking into, seed treatments, I found very few, except the idea that copper sulfate, heat treatment, and bactericides could be used with up to 90% effectiveness. But the trouble is that exposing seed to high temperature can harm the seed, potentially kill off the germination. And bactericides aren't plentiful, or I'm not even sure if they're cost effective. So that leaves, things like copper sulfate and perhaps copper, zinc, manganese seed treatments could offer some help. As BLS is mostly present on the seed surface. Do farmers want to spend money on fighting something that they may not have an issue with? Or other than that, what recourse do we have? And so, if we implemented research and development dollars towards funding long-term solutions, then could they prove fruitful within the time frame that this disease is impacting us? And I'm like, answering my own question, I would think not likely soon enough. But since the wheat genome is being mapped, perhaps solutions aren't that far away. But still, though, do we spend scarce dollars at solving something that could disappear this season due to drought or from eliminating hosts? With a dry open fall like we had this past, in 2020, many fields had significant field work done to them, and fields were worked and grass edges and sloughs were burned. Winter snow cover was sparse. If a disease that flourishes in humid, wet conditions is exposed to dry and extreme cold, will it overwinter as well as being protected under a deep layer of snow? And they're very subjective questions, and the answers I'm sure will be as well. But those are the kind of things that are going to concern me as a seed grower, and or whether to give that information to my farmer customers. And there'll be questions they'll be asking themselves. And so hopefully we'll be able to shed some light on that with some of the other professionals here on the webinar.
Thanks so much for your time today, Eric. This was great. Great to have you on, and we'll be getting some of those answers very shortly. So have a great day, sir.
Good. Thank you, Mark.
I was just about to say we will be getting some of those answers very, very soon from our researchers. Next up, we have Mira Van Berk of CCAN. I spoke with Mira as well for a few minutes last week about what she is hearing from CCAN customers in Saskatchewan and Alberta as well. So here is my short chat with Mira. Well, thank you, Mira, for joining us on the webinar today.
Yeah, thanks, Mark, for having me.
So what are you hearing on the ground in regards to bacterial leaf streak? What are you hearing from CCAN customers?
Yeah, so I remember I first heard about it at my first trade show, which was about 3 years ago. And of course, this was my first trade show, so I was kind of bright-eyed, bushy-tailed, and someone had commented on bacterial leaf streak. They had some problems. And I remember writing it down and putting a question mark. So I remember to ask some questions. And then once I started asking some questions, if people knew about it, it was, oh, you mean bacterial blight or black chaff, or there were so many different words for it. And it really was a broad spectrum of people. So you either knew about it and had been affected, or you had no idea what bacterial leaf streak was. So that was 3 years ago. And then it died down from there. And I honestly didn't really hear much about it until this past fall, when we were getting some more comments from seed growers and farmers about the damage it's had on their seed and their crop. And at first, I honestly thought it was just a regional issue, but then turns out it was a bigger thing, and it turns out it was a prairie issue. So that's kind of my experience with it.
So why is this an important topic for you guys at CCAN?
So the indication right now is that it starts with the seed, and it's a seed-borne disease. So of course CCAN is affected because our members are affected. So because we don't have a test for it, we don't know if the seed has it. So of course it's important for CCAN and our members, but of course for everybody in the industry, just to know if your seed has it.
So obviously, as you mentioned, this is something new, something people have a lot of questions about. What information are you looking for that you can give to your customers to sort of empower them when it comes to this particular topic?
Yeah, so number one, most important, is we need a commercially available seed test. So we need to know if we have it, if the seed has it, and then from there, we need to find out how to manage it. So it'd be awesome if we knew if we had it, and then once we know that we have it or don't, how to manage it and how to go on from there. And Until we have that, I think just increasing awareness with webinars like this and like listening to the professionals like Kelly, Randy, and Maria, just for building awareness is just going to make it all more beneficial for everybody in the industry.
Well, and we will be hearing from Kelly, Randy, and Maria right away. So thank you very much, Mira, for your insights today.
Awesome. Thanks, Mark.
All right, we are almost at the live portion of today's webinar. Just wanted to talk quickly with Rachel Malenka of 2020 Seed Labs, who I also talked with last week very briefly about what could be on the horizon in regards to seed testing. Enjoy our chat. Well, thank you so much for joining us on our webinar today, Rachel.
Thank you, Mark.
So what are you guys seeing at 2020 Seed Labs in regards to bacterial leaf streak?
Yeah, so here at 2020 Seed Labs, bacterial leaf streak is a disease of concern that's on our radar because it does have the ability to affect yields in wheat, barley, rye, and a particular pathovar can also affect oats. Currently, there's limited options for in-crop management. So prevention and future seed testing is definitely going to be key for controlling it. And actually, we are developing a seed test for it. So we are focusing on the pathovar undulosa. So that's Xanthomonas translucens undulosa. And then we are actually looking into the pathovar translucens as well. And if everything goes according to plan, we should have this seed test available by next season. But in the meantime, for this summer, for farmers and agronomists that are looking at their fields and are concerned about bacterial leaf streak, or they don't know if that's the disease in their field, we can do a leaf tissue test. So you can go into the field and select about 10 leaves, wrap them in paper towel, put them in a Ziploc, and then you can either ship it to our lab in Nisku, Alberta, or drop it off as well. And then from that test, we can confirm if it is a bacterial leaf streak that we're seeing or a different bacterial disease or a fungal disease, because that will affect your in-crop management options that you can use to control that disease. Yeah, so you can head over to our website at 2020seedlabs.ca. There's a subscribe to our newsletter in the top right corner, and you can also follow us on Twitter, Instagram, and Facebook. Because when the seed test does become available, we'll be sending out a notification to our customers that they can send in seed samples at that time.
Well, and this is a rapidly evolving issue, so we will be keeping up to date with it. So you can rest assured I will be checking back in with you in the future, Rachel. Thank you so much for your time today.
Thank you.
Excellent. So I'm going to be talking with several researchers right now who are going to give us the formal presentation part of today's webinar. They are live on the air with us. Rachel from 2020 Seed Labs actually is available through our chat module. So if you have a question for Rachel, please just type it into the chat box and just let her know, and she will be able to interact with you. Now, I'm going to turn the floor over to three people who many of you on our webinar today are no doubt familiar with. And they're going to shed some light on this disease, its pathology, and what they're currently doing to find out more about it and how it could be tested for. Again, if you have a question, please type it into the chat box at any time. I'm just going to introduce our speakers quickly. Maria Costanza Fleitas is a postdoctoral fellow at the University of Saskatchewan. Maria is actually an agricultural engineer who graduated from the National University of La Plata, Buenos Aires, Argentina, where she also completed her PhD studies focusing on tolerance to foliar diseases of wheat. Randy Kutcher of the University of Saskatchewan conducts research to mitigate cereal and flax diseases using integrated pest management solutions. And last but certainly not least, Kelly Turkington of AAFC in Lacombe is involved with research on the use of plant resistance for disease control in cereals, studying the impact of cropping systems on disease development, fine-tuning the use of fungicide application for disease management, plant disease surveillance and risk assessment, and the development of practical integrated solutions for crop health and disease management. Welcome, everybody. Let's all see your smiling faces if you are there.
Hello.
Hello.
Guys, how are you today?
Great.
Thank you so much for joining us. So Maria and Randy and Kelly, welcome. I'm going to turn the floor over to you guys to inform us today about bacterial leaf streak. And if at any point you guys need me, just let me know and I'll come back on screen. But for the time being, I will disappear and I will let you experts handle the discussion.
Thank you. I'm just setting the pointer here. Okay, so thank you so much for the nice introductions and thank you to the audience for their attendance. I really enjoyed the previous presentation where I learned quite a bit about the concerns of the farmers and some other organizations are trying to come up with some tools to manage this emerging issue. So today we're going to talk about bacterial leaf streak caused by Xanthomonas translucent, which is an emergent issue. But before we start this talk, I first would like to acknowledge people who authored on this presentation or who have either helped with putting some of this information together or just has provided useful information. Also, I would like to thank the organizations and institutions that will provide funding to running more research, and of course, the people who made possible this talk here today. So to all of them, thank you very much. So today, Kelly and I are going to cover 4 major topics that has been divided here in these 4 numbers. So first things we're going to talk about what's this pathogen and the disease it causes. We also gonna see a couple of pictures so we can recognize the pathogen in the field. We're gonna understand why that is so important later on in this talk, because it also occurs with some other fungal pathogens. Then we're going to move on to the management. So we are basically going to provide some basic information about what we might do to manage a problem and how it's been dealt in other places. Then Kelly is going to talk about the situation in Western Canada. And finally, I'm going to explain the ongoing and future projects. Okay, so in this slide are basically 3 images, but they're basically the same images. It's just that the one on your right has been enlarged, because I really wanted to focus your attention into these dark green areas right here on the leaves, which are the typical initial symptoms of bacterial leaf streak, which are these water sow streaks on the leaves that run parallel to the vein. And if the humidity it's really high, you might actually not be able to see the bacterial ooze coming onto. Sorry, it's not pointing. Okay, so I cannot point.
Sorry, Maria. I'll turn your red color on. Maybe that will help. Can you? Is that working? Ah, there we go.
Okay. Yeah, but I am using my mouse here, but it's not. It's just not pointing the area. Sorry about that.
Oh, that's all right. If you can, if you want to maybe direct people, just say if you want to look at the left side of your screen or the right. Of
course, yeah, sure. Of course, yeah. Thank you very much. So I was saying that if the humidity is really high, you might be able to see the bacterial ooze coming onto leaves, which are these yellow droplets right here on your on the pictures on the left. But there's also the same images and the same situation here on the picture on the right, where we can see These typical water-soaked translucent lesions running parallel to the veins, and then these yellow droplets that are onto the leaves that are bacterial exudates. Now, if the weather is right and the symptoms progress, those water-soaked translucent lesions might expand and become chlorotic and necrotic. And the leaves will eventually, here in the bottom left corner, you see a red circle enlarging this, just focus your attention on these kind of crispy leaf, which is basically how the leaf ends, like the leaf eventually dies, so the photosynthesis potential basically decreases and comes to an end. Okay, some more pictures on the left about bacterial leaf streak symptoms, but most importantly, the picture on your right, you can also see some fungal lesions occurring with the bacteria. So lots of time it's easy to misdiagnose the problem because it usually doesn't occur in isolation. And lots of cases, BLS is present with other fungal diseases as well. And here it might be a tan spot. We're gonna learn how to recognize that briefly. Another characteristic of a potential bacterial issue is the bacterial streaming, which is the bacterial cells that stream out from the cut edge of the lesion. And those are these kind of foggy and cloudy areas coming from the edges of the leaves. Basically, we can observe this by placing a small section cut from the edge of the lesion in a droplet of water, usually on a microscope slide, and we will be able to observe this bacterial issue, the bacterial cells streaming out from the cut edge of the lesion. So this is very typical from a potential bacterial issue. Okay, so I mentioned before that BLS usually occur with other fungal pathogens. I mentioned tan spot. So tan spot is a disease caused by a fungi that really start with these small brown spots here on this picture on your top left. Small brown spots that later enlarge, and then they have like a yellow halo that expands and surrounds an oval spot. So what's very typical and unique about this disease is that they develop this diamond-shaped appearance that later might enlarge and creating these blotchy areas of tan necrotic lesions. Another disease that BLS can be confused with, it's septorial leaf blotch, which is caused by another fungi, where typical symptoms starts with this picture here on the top center of the slide. You can see the elliptical brown lesions that are often surrounded by yellow halos. But something that's very characteristic is if you take a look, if you draw your attention to this picture on your top left corner, some dark fruiting bodies might appear that are called pignidia and gives kind of this speckled appearance, which is very, very typical from this disease. So Xanthomonas translucent causing bacterial leaf streak on the leaves, it also can affect the spikes. And when they are present on the spikes, they call the so-called black shaft. The black shaft symptoms are basically these dark purple streaks. If you take a look to the picture on your left, so we can see the dark purple streak coming from the glooms. And also something that's very unique from these symptoms, from this disease, when the symptoms appear on the spikes, are this kind of barber pole pattern, if you will, that we can observe in the yawns of the spikes. And I put a picture of another fungal disease, the one that's on your right, that we might confuse the bacteria, the black shaft with, which is called gloom blotch, and it's caused by a fungi. And we can see that the symptoms here involves gray, brownish blotches that usually start from the tips of the glume. And they also have, might also have present the dark fruiting bodies, which are very typical from this disease as well. So what do we know about the pathogen? Well, we know that affects most of our small grain cultivated cereals like wheat, barley, rye, triticale, oats, but also it might be present in any native weedy grasses or forage grasses. And then regarding to the environment, well, the bacteria, specifically VLS, likes areas with abundant moisture. This is because bacteria don't do well in dry conditions. In particular, VLS develops better under warm days and cool night conditions. And there's a couple of reasons of that. One is because, of course, that it's adaptive to those conditions. And the second one, it's because those conditions lead to lots of dew formation, which helps the bacteria to multiply. So the bacteria can also spread through rain splash or wind-driven rain, and that's how the bacteria blow around. But also can be spread long distances through major thunderstorm systems that basically pick up all the debris and dirt and, of course, the bacteria, and moves the bacteria around. So regarding the distribution and impact, well, we know that basically it has been reported in any region where we cultivate cereals in a big way. And I mentioned before in the symptom progress that we lose the photosynthesis capacity. So the damage of the flag leaf, and you can see here in the picture on your left, a completely damaged flag leaf. So the damage of the flag leaf is the driving factor for yield loss. Some confirmed reports of the impact on green yield are around 40%, and that comes from the United States and Mexico. But basically, yeah, we suffer from economical loss through green yield reductions. And then the grains coming from severely affected plants usually will be shriveled. Shriveled grains will have lower 1,000 kernel grains, will have lower test weights, and shriveled grains are undesirable for the myelin industry, basically because they are associated with low flour extraction rates. But also, not less important is that the pathogen might affect the balance between the nitrogen and the carbohydrate accumulation. And that means that your grain protein content might be altered too, which is an important trait in cereal, especially in wheat. Another thing that we've learned very recently is the pathogen takes over abscisic acid synthesis, which is a plant hormone that regulates the growth of the plant. And also lower seedling emergence has been also reported in highly contaminated seeds. Okay, so in order for us to manage, we really have to understand the biology of the organism. So this is the pathogen life cycle. I'm not being able to, let's see. Okay, so I'm gonna start right down here with the overwinter part. So it either overwinters in crop debris or perennial weeds, or just in native grasses, or it can survive in the seed. So seed is the most important source of primary inoculum. So when the pathogen is there and the weather is conducive, meaning like 15 to 30 degrees and lots of moisture, the bacteria is going to multiply, and it's going to be released as yellow exudates, like bacterial ooze, like I mentioned before, and splash around with the help of rain, and wind can also blow the bacteria around. So this is how they'll get onto host plants that are growing in the spring, and then they'll penetrate the plant and they'll start to form those first visible streaks that I showed you in my introductory slide. And then the bacteria will multiply if the weather is right, and then they'll splash around and infect more leaves. So if it gets to the spike, it will cause the black chaff. And okay, so it will cause the black chaff. I cannot point the spike, sorry about that. And that's how it gets into the grain and gets the seed infected. So then the pathogen keeps spreading, and it can be sprayed in the field through rain splash and wind. It can be spraying long distances in major thunderstorm system, like I mentioned before. And then later in the season, the bacteria will kind of dormant, and they'll survive in the crop debris in the field. They can survive in the soil a little bit, but mostly in crop debris, grains, or perennial weeds. Okay, so now moving to the management, let's take a look at some key points here. We know that it can survive and move around in the seed, and that's a big one. So we need to be prepared to deal with it. And in the next slide, I'm going to talk about how we might do that. The bacteria can survive in the soil, but it survives poorly. So, you know, if you have a decent crop rotation, bacteria in the soil is not going to be your problem. But if you have a short crop rotation and you have infested crop residues, when you bring the next crop in, that could be a problem. Or if you have volunteers or weedy grasses, or even just grasses watering your field, it can survive there. We know that moisture is a critical factor. So again, nobody hopes for a drought, but definitely drought conditions can help really push this pathogen back. It'll grow in temperatures between 15 and 30 Celsius degree, where there's abundant moisture. And it's really important to keep in mind that the bacteria doubles their population size every day. So every 18 to 24 hours, the population size doubles. And that's an amazing rate of replication compared to other fungal pathogens. As a result of this, sorry, I forgot to mention that this is a polycyclic disease that generates more bacteria multiple times throughout the growing season. And as a result of this, but just let me point here that fungicides are ineffective because bacteria metabolism is different than fungi, and so the fungicides do not work. And even if there are bacteria that could get an emergency use registration, what we've seen from the US just shows us that because this generation time is so short, that you have to reapply this bactericide so many times during the growing season that it's just not economical. So if I have to give recommendations for growers today, there's only a few things that they can do right now. One, it's scouting your crop and keep good records. So know where BLS show up and know which harvester grain might have the pathogen. Because if you're going to use the store grain as a seed, you want to know if it's there. And right now, the only way to know is by keeping records. We need awareness and we need to learn how to recognize it. Seed testing is on its way and it's kind of begun here, so that's going to be an option. And what we'll need in the future is staying as clean as we can from the start. And this is going to be a really key piece of management in this disease. So really avoidance what we want, because we just don't have many management option tools. We will use host resistant if it becomes available, and we might get some incremental help with seed treatment if they become available as well. But like I said before, they only can give some incremental support for management. They won't solve the problem 100%. We're really going to have to rely on clean seed programs to help to manage the disease along with crop rotation. So for the future, we should watch and scout for it. We need to learn how to recognize it. This is a new disease for most of the people. So really, we really need to know how to recognize it and learn where you can get some diagnosis support if you're suspicious that you have an issue. Again, we need awareness and recognition, like Mira mentioned before. And lastly, crop rotation, again, it's not going to solve the problem 100%. It'll just provide some incremental help. But I'll just leave a question mark right here, because that doesn't mean you shouldn't rotate. I mean, you should always practice a good, long, and diverse crop rotation for the best of the farm unit for lots of reasons. Bacterial leaf streak is just another one on the list. And now, Kelly, the floor is all yours.
Thank you, Connie, for that. So what I'd like to do now, oh, sorry, Mark, I'll let you introduce me or just continue.
Yeah, I think I read out your bio earlier,
Okay,
Kelly,
excellent. So
welcome to our webinar, and we look forward to hearing from you.
Very good. So the component of the presentation I'll be covering is to give you a brief overview of the situation in the prairies. And if you look at development in the northern Great Plains, it was really in the mid to late 2000s where in North Dakota, South Dakota, and Minnesota, they started noticing issues with it. And unfortunately, it has continued to develop into more of a sort of an ongoing problem. So they're looking at screening material, the commercially available varieties in terms of reaction and so on. But if we look at the prairie region, and we'll turn our attention to Manitoba first, and the information here is primarily from 2020. There was one confirmed case based on lab testing from last summer. There were other anecdotal reports. Some of the symptoms were misdiagnosed earlier on and likely were confused with the leaf spot complex, which Connie has introduced. So that would be comprised of tan spot and the septoria complex. It had a limited sort of area that it affected, and it appeared to follow along some significant and severe weather events, and we'll touch on that in a few moments. It was primarily reported on one cultivar, AEC view field, but certainly other cultivars are susceptible. And if you look at the slide here, unfortunately you can't see my pointer, but you look at the rainfall data in 2020 for May, June, and July. There were areas around that sort of broad region that had some pretty significant rain events. And as Connie has mentioned, severe weather events are likely a risk factor, whether it's thunderstorms, hail, driving rain, strong winds, and so on. Those all help to create wounds on the wheat or barley plant, which then the bacterium can access in order to infect the plant. So certainly here it illustrates that appearance in Manitoba in 2020 was associated with some significant rain events during the summer. If we look at Saskatchewan, the situation there, there may have been some misdiagnosis in the field. There was one confirmed report of the issue in terms of the Sask Plant Health Lab, as well as other anecdotal reports, and affecting a number of different varieties. If we turn our attention to Alberta, and I'll talk a bit about some information from Mike Harding, Ji Fang, who's with the Alberta Plant Health Lab, and our own experience over the last 20 to 25 years. Certainly the issue of concern is Xanthomonas translucens pathovar angulosa, and that's work that Mike Harding has done along with Ji Fang, and then a colleague with Ag Canada, a taxonomist in Ottawa, James Tambong. In 2012, our lab in Lacombe received a number of samples from southern Alberta where the grower had sprayed 3 times, so at herbicide timing around flagleaf, and then anthesis timing with little effect or no effect really. So the concern was initially fungicide non-performance and potentially the appearance of fungicide-resistant strains of different organisms responsible for the wheat leaf spot complex. We did some bacterial streaming tests, and all of the samples showed very significant streaming, indicating a bacterial issue. And that, along with the lack of fungicide performance, would certainly indicate that it was a bacterial issue. In 2013, in southern Alberta, Mike reported the disease in a number of fields. 2017 and 18, there were a few more fields that were severely impacted. We were out in the Castor area at a Applied Research Association field day in 2018 and noticed actually some very severe BLS symptoms in barley and some yield trials there. Mike estimates in southern Alberta in 2018, they were probably looking in some fields that were severely impacted, looking at fields that had between 20 to 30% yield loss. Next, we come into 2019 and 20, certainly 8 confirmed fields, primarily in southern Alberta. 2020, we did have a report and a confirmation through Ji Fang's lab of both Viewfield and Brandon being affected in a particular farm east of Edmonton. And we've had dozens of anecdotal reports. So far in 2020, the issue, again, largely reported on AAC Viewfield, but certainly reports on other varieties. There's certainly some evidence of seed transmission, and this is based on the observations over the last few years, as well as the nature of the disease itself and the critical role that seed infection plays. Overall, though, if we go back to the mid-90s, we have observed bacterial issues in a range of cereals, whether it's wheat, barley, or oat. Often these are isolated. So in our screening nursery, we might have several thousand breeding lines we're looking at for scald resistance in barley. We might find one or two of the hill plots with symptoms. So really more of a curiosity. We would see that in some yield trials and occasionally in some commercial fields. So it's an issue that is certainly appearing to become more and more of a concern. Certainly started off as more of a curiosity and something that wasn't really very common. That's obviously changed over the last 5 to 10 years. And I think the next, so this is just showing you the situation in May to June and in July in Alberta, and again showing you areas where it developed in southern Alberta and sort of east of Edmonton. We certainly had some pretty significant rainfall events, and certainly from what Mike has indicated, thunderstorm systems and so on, which are a risk factor for BLS appearance and development. Mike also did some excellent sort of preliminary work looking at virulence tests. So we have a lot of questions regarding variation in Canadian varietal reaction. So Mike did focus mainly on wheat, set a number of CWRS varieties, CPSR, CWSP, CWRW, and one Amber Durham, as well as a single barley variety. And the testing that Mike did at Alberta Agriculture and Forestry in Brooks indicated that it was the pathogen was virulent on all, and really not a lot of variation. Everything was more or less susceptible, unfortunately. So with that, I'll turn it back to Connie, and Connie will talk a bit about some of the ongoing and future projects that we're looking at.
Thank you. So yeah, coming back here, I'm going to talk about a little bit the ongoing and future projects. So in my next slide, I just have listed a number, well, our collaborators that are running with more research and assistance to help us get some things figured out with respect to this emerging issue we have right now in our cereal production in Western Canada. So this is a 3-year project that we recently got funded to work on barley. And we basically are aiming to establish a seed testing protocol and an indoor base disease evaluation method to improve the management against BLS, particularly working on barley. In this project, we, oh, sorry, animation. So we cannot do animation here because there was a picture behind that was showing how the process starts. So our first objective is to establish a seed test protocol to detect pathogenic strains of Xanthomonas translucens, because not all of the pathovar are pathogenic on our cereals. But we basically, wanted to develop here a simple and rapid seed test protocol using a novel technique called LAMP for barley seeds. Briefly, I'll mention that the protocol consists of a visual detection method, and by visual I am meaning a colorimetric reaction that can detect the kernels infected with pathogenic Xanthomonas translucens that cause disease on cereals from those that cause disease on non-cereal hosts. So a seed testing protocol to identify contaminated seed is will be one of the most effective and sustainable tools for BLS management. And we can see here that we basically do we obtain the DNA of the bacteria from samples, from grain samples, and we run the PCR analysis, or LAMP, like I mentioned before. And then the results look exactly like the picture that I'm showing you here on your right, which are these tubes right here, where the yellow color indicates positive. That means that the seed has the strains that are pathogenic on cereals, whereas the ones that are pink color are strains that do not cause disease on cereals. I'm gonna skip this. So, okay, for our second objective, we aim to establish an indoor disease evaluation method. And this is basically will help us screen barley lines in the future. We plan to conduct a series of experiments under controlled condition, and we will test barley line with different pathogenic strains, including like different inoculation methods, different evaluation disease methods, so we can standardize a screening protocol to screen lines in the future. Yeah, like I mentioned, this indoor testing protocol will be used to screen commercial cultivars to find source of resistance that will help us identify germplasm for breeding programs. And since we're also going to include some wheat lines, we're going to test as well some possible cross-infection with wheat. Another part of the objective too is to determine the virulence of the pathogen. So basically how aggressive is each isolate. So in here, we have basically 4 different isolates just for the purpose of the illustration. And we can see here that isolate B, once we inoculate a collection of cultivars, we can see that 4 spikes out of 5 are affected, meaning that this isolate is more aggressive, as we compare with isolate C, where we can see that from all the cultivars inoculated with isolate C, only 1 spike out of 5 are affected. So they will kind of give us a better insight regarding the virulence and aggressiveness of the pathogen, and will help us understand the impact and the pathogen characteristics. And the final objective of this recent funded project is to determine BLS prevalence strains from strains isolated from Canada. So basically in here, we wanted to know what we have in Canada. Are our isolates different from the US? What's going on here? So for this study, it is crucial for us to collect a large number of isolates that we can analyze and sequence the genome and see what we have. Here, basically, we're going to characterize different pathogenic strains across Western Canada using molecular techniques, like I mentioned before. We also gonna find out if there's any predominant genotype in the region. So yeah, these information will indicate the extent of the problem, not only to pathologists, but also to agronomists, breeders, seed industry. And this work is very, very important. It's foundational because it provides some specific pathogen detection protocols. I mean, it will help us validate our seed testing protocol with specificity. And lastly, or another project that has recently submitted is to work on wheat, bread and durum wheat, where we basically aiming to do similar investigation and experiments for barley. We want to develop a standard seed test. We want to study prevalence and virulence. But most importantly, we're also going to assess the variation of host susceptibility. So basically here we will screen indoor lots of commercial varieties that include bread wheat, durum wheat, oat, and triticale for resistant to BLS. And basically the aim is to identify potential resistant or less susceptible genotypes, if you will, to pathogenic strains of Xanthomonas translucent. And then Kelly, with the aim of increased screening capacity, will also establish a field nursery in Lacombe, where the weather conditions are more conducive for BLS development. So that's really, I guess, what I wanted to cover. There's lots more we can talk about, but in the interest of time, I'll leave it right there. And thank you. I hope you find this information useful.
Thank you so much, guys, for that great presentation. Very informative, to say the least. We have a few minutes to take some questions from our audience. So if you have a question, please type it into the chat box at any time, and our speakers will try and answer it for you. Again, Randy and Kelly and Maria are available to take your questions. I had a question, guys, regarding the variety AAC view fields. There was a comment that AAC view field has become the most popular HRS wheat grown in many areas. Could that be one of the reasons why it gets associated so much with bacterial leaf streak?
I'll maybe jump in there, Mark. It's Kelly
Sure.
here. You know, there is some analogy. I don't know, some of the audience members may remember the tip ripening that occurred in AC berry way back in about 1999. It was associated with some significant weather events towards the latter part of June, early part of July, and berry was fingered as being an issue, but it did occur in other varieties, so there could certainly be a component of that. You know, the challenge, especially with BLS, especially where the situation or the issue is just starting to become more of a problem, is that seed-borne inoculum will be an important source of that particular pathogen. As it becomes more established on crop residues, and that's likely the case in Minnesota, North Dakota, and South Dakota, crop residue becomes important in addition to seed-borne disease. So certainly that could be a factor in terms of its appearance, in terms of it being a very popular variety, and you know, you're seeing more acres of that variety, and thus you're exposing that variety to the pathogen more over a large geographic area. Same thing with AC berry. It grown widely across the prairie region, so it just happened to be a common variety that was exposed to some of these sort of detrimental weather conditions towards the latter part of June, first part of July.
How does the seed-borne isolate cause leaf disease? I think you touched on how some of that works, but can you go over that again on how the seed-borne isolate actually causes disease in the leaves?
Oh, so how the seed-borne pathogen causes disease on the leaves? Is that what you're asking?
Yes.
So, okay, so like I mentioned before, if the pathogen it's in the seed, we still don't know whether it is in the surface or inside the seed. That's something that's not clear yet. When the plants germinate and then the pathogen is in the seed, and if the weather is conducive, and I mentioned before that the pathogen grows between 15 and 30 Celsius, and there's moisture there, the bacteria will start just multiplying. And there will be some release so the bacteria will be splashed around. And we mentioned before that wind can blow the bacteria, but also this will be just only for short distances. Thunderstorm system might move the bacteria through long distances. And then we'll reach to the leaves, and then we'll start causing those water-soaked symptoms that I mentioned at the beginning. And then how's like if the weather is right, the bacteria just keep multiplying. I mentioned before that it was a polycyclic disease. That means that generates more bacteria multiple times. And then if we get to the time of heading and the disease is still progressing, they will get into the spike and they will develop these black shaft symptoms. And that's how, again, the seed gets infected, and then the cycle starts all over again.
What is known about the stomatal size, and does that limit the infection?
Well, it's been some research conducted about there, but not only the stomatal cells are a key point of entry of the bacteria, but also natural wounds caused by hail I'm not quite sure about the stomatal size right now, but if you want, I can take a look, like, and we can have a discussion later. Yeah, because I don't recall right now any study about the stomatal size and bacterial leaf streak affection.
And Maria's contact information is available in the presentation, which will be posted to our website. So if you'd like to get in contact with Maria, I'm sure she would really appreciate hearing from you. In regards to seed treatments, is it correct that those are not very effective on the seed-borne pathogen?
Well, in this case, for bacterial leaf streak, there are not effective seed treatments right now. Like our first presenter mentioned before, there are a couple, but that won't solve the problem 100%. We need to come up with an integrate management where we use crop rotation, clean seeds, resistant varieties. So resistant varieties won't solve the problem again 100%. So we need to have like different tools, because let's say that you do crop rotation, but then you bring the pathogen with the seeds, or let me think, or let's say that you have a resistant variety, but then the crop debris, you have so many bacterial cells relying there that you still have the problem in the next crop season. So there's no black or white answer right here. Like, we need to do several things. We cannot rely only on one tool. And I read there in the question that some people were asking, like, if we're going to, like, once the seed test is developed, like, if there's any threshold that we should establish, because that's another thing. Like, if we have the seed test, and then let's suppose that these become a major problem, and we have several lots infected with the bacteria and seeds infected with the bacteria, we need to come up with a test as well that kind of tells us the risk, like how much do we have and what is the potential damage. There's some studies that has been indicating that there's a threshold, like there's a percentage of contaminated seeds that will actually become infected plants in the future, but that's some studies that still needs to be conducted. I mean, we learn a lot about our U.S. neighbors, our American neighbors, because it's been an emerging issue quite for a while there now, but there's lots of things that we need to do here in Canada as well.
Mark, it's Kelly here. Just to follow very quickly up on what Maria said, if you go back and look at some of the work done by Shad and Forster, I think primarily in Idaho, but they looked at threshold levels for seed, and their focus was more on washing the seed with a known volume of water, and then looking at the number of colony forming units per volume of that seed washing, and their threshold was about 1,000 colony forming units in that seed wash. Their information also indicated that this was roughly equivalent to a seed infection level of 0.014%. So in terms of seed testing, we may be moving more in a direction of maybe seed washing and then looking at the colony forming units per volume of fluid per unit weight of seed. So yeah, it's looking at some of these seed thresholds, it's rather disconcerting, quite frankly.
Once a test is developed, what level of infection would be acceptable for use as seed?
Well, that was I was addressing previously. Like,
right.
There's more research that need to be conducted after that. Like, what would be the acceptable threshold? We need to find out. Like, there's some study that has been done, but previous authors that report that some plants get infected from, let's say, if we have a lot with 3% of contaminated seed, not all the plants will result infected. But like I said, that depends on the weather. If the weather are conducive and you're getting these wind and rain splash, and the weather is right, meaning that the temperature that I mentioned and moisture is there, then you'll still have the problem because just the bacteria just have an amazing reproduction rate compared with fungal pathogens. So we definitely need to run more research to define that in the future.
Mark, it's Kelly here. Just a really quick comment, and it relates to the first speaker. I've sat on both the wheat rind triticale and oat and barley disease evaluation teams as part of the Prairie Recommending Committee committees for both those sort of broad crop types. And certainly the committee takes direction from what's happening in terms of the situation in farm fields, what the breeders are telling us, and so on. So in terms of bacterial leaf streak has never been a priority 1 disease, so it's not something that we routinely screen for. Over the last probably 10 to 15 years, we've seen a desire to sort of focus on more key issues and less so on more minor issues. A good example of changes that do occur is in relation to stripe rust, and that originally was not a priority 1 disease. We had outbreaks of it, especially in southern Alberta, and certainly since probably the 80s, even before that. But as stripe rust became more of a concern, we were routinely seeing it on our surveys. The pathologist proposed that we would change the
Are you still there?
process wasn't immediate, but it shifted over the years. So certainly, this is something that we'll bring up with the disease evaluation teams to look at this issue and whether we can justify resources in terms of screening for it and potentially adding it to the priority 1 disease list.
Before we sign off for the day, I had a question regarding forage grasses. Other pathivars of this infect forage grasses. What's the situation with those in Canada?
Okay, so yeah, the pathovar, it's a very complex situation that happens with the bacteria. They're basically determined by host range, molecular, and biological characteristic. So we do know for sure that the translucence groups are the one that cause BLS on cereal crops like wheat, barley, rye. And then we have the Graminy group that has 5 pathovar. One has been listed very recently. And when the disease is there, it's called bacterial wilt, which is the pathobars of Xanthomonas translucens that affect non-cereal host. So if by forage you mean like the forage barley, I always read about the barley that goes for the malt industry. But if you are referring to the pathovar affecting barley as a forest, there might be the translucent group. And we have right now 5 different pathobars in that group, which is undulosa and translucent. But there also are cerealis, ordae, and secalis.
Mark, it's Randy here. I just maybe make a comment that you ask a lot of good questions, or the audience does, and I think you all realize now there's a lot more questions than answers. So, I mean, we know generalities that bacterial pathogens, they either tend to be vectored by things like insects or they're passive. And so in this case, it seems it's passive because it's associated with storms and things that wound plants. So stomatal penetration may be irrelevant or less important if it's wounding. Seed to seedling transmissions, another big question. So how much infection on the seed is too much? We really don't know. I guess we need to know what is the seed to seedling transmission. So if you look at the pulse crops and some of the diseases, in chickpea, it's very important. There's a high rate of seed to seedling transmission if you have ascochyta on the seed. Whereas in pea and lentil, it's much, much less so. And then it's affected by weather. So like Connie was saying, it may vary between, say, the northeast Saskatchewan or Manitoba, where it's damper and maybe a bit warmer sometimes, than, say, southern Alberta, where it may be damp, but it may be cooler. So these are lots of great questions. And unfortunately, we can't really give you definitive answers right now. That's what we're working on, things like this.
That's true. And someone mentioned in the chat that we should be having an international summit on this topic, which is a terrific idea, actually. So hold that thought, because that might be a great thing to organize for the future. So thank you, everybody. If we didn't get your question, this will be available on our website. You'll receive an email with a link to the recorded version. So feel free to get in touch with our speakers. Thank you very much, Kelly and Maria and Randy, for your time today. And thank you to Eric and Mira and Rachel as well. And thank you to our audience for spending your lunch hour with us. I also want to take the opportunity to thank our sponsors, 2020 Seed Labs and CCAN, for their support. And again, this whole webinar will be available on our website, germination.ca, very soon. Again, if you registered for our webinar, which you all did as you're all on here with us right now, then you will get an email with a link to the recorded version, which you will be able to watch on our website. So thank you, everybody, for joining us today. Take care, Kelly and Maria and Randy. I hope you guys have a great day, and we'll talk with you all soon.
Thanks, Mark.
Thank you.
Bye for now.
Thank you, Mark.
Thanks,
Bye-bye.
Kelly.
Thank you, Mark. Bye.
Thanks, Maria. Thanks, Randy.